Right, so this video will be about phylogenetic trees, how to make them, and what they mean, how to interpret them. A phylogenetic tree is just looking at different organisms and see how they're related, just like your family tree, really. You can see here in a very simplified version that the main branches of the tree are the bacteria, the eubacteria, archaeobacteria, and eukaryotes. We are part of the eukaryotes in this animal group right here. And one thing what I wanted to point out is that it's quite interesting that most of these organisms are invisible. Only a very few of these animals, the fungi and the plants, are actually visible to our eye. So don't be surprised, but most of this world out there is unseen and unknown to you. But how do we get to a phylogenetic tree? We can't just look at the organisms and compare them. That's what they did in the old days. And Carl Linnaeus in Sweden, he just looked at all different organisms, looked at numbers of hands and feet and vertebra and different parameters. And so he started classifying these organisms. But it's gotten really complicated, especially with the organisms we can't see. So nowadays, most of the classification is based on genetic material, and they're using 16S or 18S rRNA genes to compare and classify the organisms to see how related the different ones are. And that's what we're going to do in our activity today. We are trying to build phylogenetic trees. There are a couple of different programs, fairly simple to use, where you can build your own phylogenetic tree. One of them is Phylogeny, which was created by the EMBL in Europe, and they programmed this database. So it's fairly easy to use. You just type in your names of the different organisms you want to compare, and let's do a couple of different organisms. I'm not sure if you know what a mus musculus is, but it's actually just a common house mouse. And you enter it, it gives it a code, and then you keep going. So the next one is Caenorhabditis, which is a nematode. And we're gonna pick a couple of maybe a plant. Arabidopsis, which is in the mustard family, a small plant, commonly used for research as well. Let's get some bacteria. Staph aureus, our friend. Gram-positive bacteria, E. coli, gram-negative bacteria, and a couple of other ones. Let's get Helophorax. And what else? Let's get a chloroplast genome. Cyanobacteria. Okay, so let's see what we have. A couple of different organisms let's make sure we have the different groups. And so we've got a bunch of different organisms, and we generate a tree and we visualize in iTool. Alright, so this is one way of looking at it. There are different ways how you can look at the tree as well. You can do a circular, you can do a rectangular tree, you can do an unrooted tree. So there are lots of different ways of how to look at it. But we're gonna compare it a little bit. But you can see so how the longer the lines are, the more distantly related organisms are. So again, Halopherex and Haloraptors, they're very close related because they're both archaeobacteria. E. coli and Staphylococcus are close related, and cyanobacteria because they're all eubacteria. And then we have Arabidopsis, which is a plant, our worm and our mouse, and so they again group into a different group. So it makes total sense how the tree pans out. Let's look at a couple of other methods of how to do a phylogenetic tree. And these ones are based on 16S sequences. Again, this one is phylogeny.fr, and we are going into the regular one-click mode, which is a simple pathway, and we're just using it's not working. Here we go. So I'm just copying and pasting different sequences, 16S, 18S sequences, into the program, separated by different formats, and then we see what we get on this one. Okay, so I'm copying the sequences from our worksheet, pasting them in there, and you can see they're all formatted like this. And you just need to say submit. And let's see what we get. So first the sequences are all aligned, and then this is the alignment process, and then there's curation, and then there's phylogeny development. It's pretty cool how this works behind the scenes. So you don't need to know any sequencing or any programming or any of these other skills and tools. You can just use these pre-made tools that are ready for you to use. So this is our tree according to phylogeny.fr. Again, we have the house mouse, we have a worm, we have humans, we have chimpanzees, we have an unknown organism in here, we have our archaeobacteria, we have our chloroplast, our cyanobacteria, and a couple of other ones. So it's quite interesting of all the results what you can deduce from this tree. For example, obviously all the higher developed organisms are grouping together, the archaeobacteria grouping together, so our unknown from this result, you would know that this is an archaeobacterium as well. Also pay attention to the chloroplast here. So the chloroplast genome are related to cyanobacteria, and it's thought that the chloroplast and mitochondria were free living organisms, and through the endosymbiont theory, through the endosymbiont process, they merged with another eukaryote and then became sort of a endosymbiont organism. So our mitochondria and plants' chloroplast, they still have their own genetic material, which is traced back for the chloroplast to cyanobacteria. And then we have our bacteria, again falling into gram-positives and the gram-negatives as well. So again, makes perfect sense, really simple. And I'll show you a third method of doing this. The phylogeny.fr is a great program, but sometimes a bit finicky in terms of processing. I'm also going to show you the EMBL, European Molecular Biology Lab pathway to do the multiple sequence alignments. So again, you do multiple sequence alignments, and again, there's lots of other tools, what you've seen at the NCBI as well. And here's our phylogeny one. And here I'm going to just paste our sequences again in faster format. And you can use an example, but we just didn't do our own ones. And that didn't work. So let's align them first. So this is the alignment, and then this is the alignment result, and now we need to just find the tree. Here we go. Here is our tree, and again the tree looks slightly different, but I think the bottom line should be very much the same. So again, the different methods of how to make a phylogenetic tree. You get the ITOL, and we just put in the names of the organisms. You use the phylot, phylogeny.fr, and again you put the 16S, 18S sequences in it, and then you're using, here we go, our EBI program, and you get the sequences as well. Again, you can play around with what it's supposed to look like, and you can do colors and all sorts of other things and really compare. So think through it, see what it means. Does it make sense? Why are the different groups grouping together? But really great tools used all the time in research to figure out how organisms are related. Alright, so this is our phylogenetic tool, and there are different ways of making the phylogenetic trees, but I want you to make sure you understand how to make them and also how to interpret our phylogenetic trees. How are we related? How are you related to a mouse and a bacterium? See you soon!